So the first step of action potential is the stimulus. A stimulus causes the inside of the cell to move towards negative 55. It starts at around negative 65 or milli MV. The stimulus can include a light stimulus, a photoreceptor, a mechanical stimulus to the skin, or a stretch to a muscle receptor. The greater intensity of the stimulus, the greater the number action potential is, and then the action potential begins.
Secondly, the voltage-gated sodium channels open. The sodium diffuses into the cell and cell membrane and then depolarizes. The membrane eventually gains a positive voltage. That current travels rapidly through the cytoplasm of the axons, insulated by the myelin sheath, until it reaches the next node of Ranvier.
So these are the nodes of Ranvier, right in between these little guys.
Yes.
Those are the nodes.
Those are the nodes in the space and where the action potential and the
this
impulses.
whole thing is the axon, and this is the myelin sheath.
Yep.
So during repolarization, the potassium diffuses out of the cell. The cell's voltage is lowered to the resting potential of negative 70 when it started at negative 65. So it's a little bit lower than it was when it's at the first initial resting potential. And then the voltage-gated sodium channels close, so there's no more positive charge that enters the cell.
Next, potassium channels take a long time to close. The excess potassium is let out. The membrane potential goes below the resting potential to negative 80 MV. After that phase occurs, the next cell goes to the highest voltage from depolarization. The purpose is to restore the resting membrane potential. Lastly, this is the ready state of the cell, a process that's balanced by ion leakage. And after this and the ion pumping without any outside influence, it will not change. To get an electrical signal started, the membrane potential has to change and go back to some sort of resting. After being fired from previous action potentials, the membrane returns to its resting state by the sodium-potassium pump, which balances things out. All of the gated potassium and sodium channels are now closed.
They're all closed, and it's back to its resting state.
To go more into further detail in the anatomy of a neuron, we have this. These are the synapses, the waves of electrical impulse, and in here are the nodes of Ranvier, which is kind of the space in which the electrical charges kind of move through. This is the myelin sheath kind of protecting the axon. We see some vesicles, the gap junction in which there's like a little bit of a space and connects from one neuron, as you can see with this paper clip, like a receptor to another receptor binding. Up here, these beads you can see are neurotransmitters connected to our dendrites. Speaking a little bit on diffusion. Diffusion is basically the transition of electrical impulses or electrical charge between neurons, and from each exchange there's also chemicals exchange. For example, like the sodium kind of diffusion is moving sodium from one pump to another, moving it from kind of across membranes.